Bismillahirrahmanirrahim. Today I would like to share the knowledge about protocol of ELISA. My name is Sarfraz Rahman. I am a PhD scholar in the Department of Parasitology. I would like to share this knowledge with references to these books, The Cloning and DNA Analysis, 6th edition by T.A. Brown, and this one is the research paper, A Short History, Principles and Types of ELISA and Our Laboratory Experience with Peptide and the Protein Analysis using ELISA by Suleiman Ahadin. The third one is the internet sources, and you can see that these two ELISA plates, this ELISA plate has the two different colors that actually shows the positive or negative sera, and similarly you can observe in this ELISA plate it has also the two different colors which actually shows the positive or negative sera. Please subscribe my channel and also push the button of bell icon. So I would like to tell about the ELISA. It is actually abbreviation of enzyme linked immunosorbent assay, and it is also called the enzyme immunotest, or that is also called the enzyme immunoassay. Its short form is EIA, and this is the main principle of ELISA. It shows the antigen antibody reaction, color changes, and this color changes it actually serves to identify the presence and concentration of molecules in biological fluids, and by using the enzyme linked conjugate and enzyme substrate in 1960, that RIA, that is the radio immunoassay technique. It was discovered by Yellow and Burson. They actually used to measure the endogenous plasma insulin level. And the ELISA term, it was coined in 1971 by Engvall and Perlman. They belong to Stockholm, and this picture it is actually the Peter Perlman, and this picture is actually the Eva Engvall. So ELISA, it is actually can be used to detect both antigen and antibody, and it is very sensitive. It can be measured picogram per ml and relies on totally on the monoclonal antibodies, and it may be run in qualitative or quantitative format. So this one is the principle of ELISA. So it actually shows the antigen antibody reaction through the color changes obtained by using an enzyme linked conjugate and enzyme substrate, and that serves to identify the presence and the concentration of molecules in the biological fluids. This is the picture, actually it shows the principle of ELISA, and you can see this is the well of the ELISA plate, and these are the antigen that is below the in the bottom of the well, and these are the antibodies, and then you can put the the secondary antibodies. It is actually conjugated, and then you add the substrate, and that will combine to the enzyme, and it show the color, and this color it actually can be measured the OD value with the help of spectrophotometer or ELISA reader. So the ELISA labels, it is actually used to detect the binding of antigen antibody complex and have a high specific reactivity, easily coupled to antigen antibody complex, and must stable and enzyme used in labeling that should not normally present in the patient samples. And enzymes are actually used to that mainly the beta galactosidase and that the glucose oxidase and peroxidase, alkaline phosphatase, that alkaline phosphatase and p-nitrophenyl phosphatase used as a substrate and that produce a yellow color in the positive reaction. Peroxidase conjugate 5-amino salicylic acid and that is the orthophenylidiamine are used to that produce the color that is the brown color, which is it actually show the positive reaction. And beta galactosidase that is used the sample it actually must be read in a fluorometer. This is the picture. You can observe that first one is the capture antibodies, then antigen, then detected antibodies, then the conjugate. It actually the secondary antibodies. It has the enzyme, and this is the signal when you add the substrate, and this enzyme produces the color, and that is the signal. So the stages of ELISA, the absorption of either antigen or antibody to microtiter plate, the addition of the test sample and subsequent reagents, the incubation of reactants, formation of antigen antibody complex, and the separation of bound and free reactants by washing, the binding of enzyme to the target antigen or antibody, the addition of the substrate, production of a visible signal, and the visual or the spectrophotometric reading of the assay. And you can see that in this picture, that is the first step, that the antibodies, then washing, then antigen is added, then washing, then secondary antibodies are added, and then the substrate is added, and it will produce the color. The types of ELISA, basically it is divided into the two main categories, that the homogeneous ELISA, it is for the spatial use, and while the heterogeneous ELISA, it is for the general use. And it has the main four types, that direct ELISA, indirect ELISA, sandwich ELISA, and competitive ELISA. The direct ELISA, in which you see that the antibodies, and then indirect ELISA, that is the antibodies, and sandwich ELISA, you can observe the antigen, and in competitive ELISA, you can see that the antigen or antibodies can be observed in that competitive ELISA. So the types of ELISA, the two general headings, the homogeneous enzymatic immunoassay method and heterogeneous enzymatic immunoassay method. The homogeneous enzymatic immunoassay method, that the enzyme become inactivated when they are bound to the antibody, and it is very important that there is no stage of washing where the antigen is separated from the medium. So it employed to measure the substance in small quantities like the therapeutic drugs, and it has the disadvantage, it is expensive, or it has the low sensitivity, while it has the advantage, it is easy to use. And this is the diagrammatical presentation. You can see in that picture that this is the antigen, it is captured between the two antibodies, and that can be easily measured with the in this curve. So the types of ELISA in this that the second category, that is the heterogeneous enzymatic immunoassay method. It is more commonly used and prevent interference of any molecule in the medium with it after the binding of the antigen and antibody. It will make the antigen antibody complex. And in this category, that in this method, the washing stage is very important. That it actually used to separate the binding antigen from the free antigen after the antigen antibody interaction. It is more sensitive than the homogeneous ELISA. ELISA is the heterogeneous immunoassay technique. It is used to detect the specific antibodies and soluble antigen. And this is the picture you can observe in this picture. That is actually this one is the antibody, or this is antigen, then antibodies that is conjugated, and it shows the signals. So the direct ELISA, it is used for the antigen screening. It's developed in 1971 by Engwall Perlman, and determined the amount of high molecular weight antigen. And surface of the plate is coated directly with the antibody or antigen, and enzyme tagged antibody or antigen enable the measurement. Enzyme labeled primary antibody is used. Secondary antibodies are not needed in this type of the ELISA, and it is used and it can be measured the antigen of interest. In this picture you can see that first one is the antigen, then the primary antibody that is conjugated with the enzyme. Then the indirect ELISA, it is developed in 1978 by Lindstrom and Weger, and it is actually used to determine and separate the antigen to be measured is not the primary antibody, and diseased serum is added to the antigen coated valves, and the plates are incubated. Incubation, the antibody formed against the antigen is in the diseased serum. Plaques produce an antigen antibody complex. A secondary antibody that recognize the antibody in the serum, and that is tagged with the enzyme is added, and the substrate of the enzyme is added to the medium to produce color, and the concentration is determined. And you can see in this picture that is the antigen, then primary antibodies if you added the serum, and the secondary antibody that is tagged with the enzymes. Next one is the sandwich ELISA. It is also used for the antigen screening test, and it is developed in 1977 by Cato and his co-worker. Valves are coated with the captured antibody, and blocked samples is added to the microplate valves coated with the antibodies. Plate is incubated for some time and washed. The washing remove the unbound that the antigens. When the antigen specific to the bound antibody is found, these antigen cannot be removed. Antibodies that are tagged with the enzyme specific the antigen are added and incubate, and after the incubation and washing, if there are antigen in the medium, these cannot be removed as the enzyme tagged antibodies are bound to them. Enzyme substrate is added to the medium, and coloration is ensured. This coloration actually shows a positive result, while the lack of the coloration indicates the lack of the enzyme or negative results. As the relevant protein is stuck between the two antibody molecule, and this method is called the sandwich ELISA. In this picture, you can also observe that this one is the captured antibody and this one is the antigen, then detected antibodies, then the secondary antibodies with that is the conjugated. Next is the competitive ELISA, and it is actually the antigen antibody screening test, and it is developed in 1976 by Yorde and his co-workers. The surface of the well is coated with the antigen specific antibody or antibody specific antigen. The samples to be measured and enzyme tagged antigen or antibody are placed into the well simultaneously. The tagged and untagged antigen, the patient antigen or the antibody molecule compete to bind to the antibody or antigen in the wells. After the wells are washed and enzyme substrates is added, the resulting coloration enables the quantifying the concentration inverse proportion between the analyte that the concentration and intensity of the resulting coloration. Put it and differently, when the amount of the antigen or antibody analyzed in the serum is low or high, absorbance is obtained. The greater quantifies that produces the low absorbance, and this is the picture and it actually shows the that this is the antigen or this is the antibody that is conjugated with the enzyme. This is another antigen and it is detected by another antibodies that is conjugated with the enzyme. This is the picture in the direct ELISA. This first picture you can see that the antigen is coated and then enzyme conjugated antibodies is added, and this will show the color. While in indirect ELISA you can see that the first one is antigen, then that the primary antibodies, then you see that the secondary antibody is added where there is conjugated with the enzyme and it will produce the color. While the sandwich ELISA, you can see that the first one is the antibody is attached, that is the captured antibodies, and this captured antibodies when you add the antigen, it will attach to the captured antigen, and then you should add the another secondary antibodies which is conjugated, and it will produce the color. While in case of the competitive ELISA, you can see that these are the antigen which are attached with enzyme, and another antigen without that having the antigen. These antibodies actually have the antigen, and this antibody that have the no antigen, and these two have compete to each other, and it will produce the color. If it in this ELISA, its phenomena is totally different. If the OD value is high in this case of competitive ELISA, that will indicate the negative sera, while any sera has the low OD value, that actually show the that the positive in this case of the competitive ELISA. So requirements of ELISA: the purified antigen if you want to detect or quantify the antibody, purified antibodies if you want to detect or quantify the antigen, and standard solutions, positive and negative controls, sample to be tested, and micro titer plates, and the plastic trays with small valves in which the assay is done, then the wash fluids or buffer, enzyme labeled antibodies and enzyme substrate, and then you need to perform this ELISA, that ELISA reader. It is very important to quantify, to observe the OD value. And for quantitative measurement. In this picture you can see this is the performance of the ELISA, and this is the ELISA reader. So the advantages and disadvantages of the types of ELISA. and there is 4 types: direct ELISA, indirect ELISA, sandwich ELISA, and competitive ELISA. And in case of the direct ELISA, it is actually show the antigen screening, and that the false positive results, and it has the advantage that low sensitivity. While in case of the indirect ELISA, it show the antibody screening test, and but it has the disadvantage cross reactivity, extra incubation steps are in this step. While in case of the advantages, that you can observe that the increased sensitivity and the wide variety of the labeled secondary antibodies are available commercially, and maximum immunoreactivity of the primary antibodies, use of the different visualization markers. And in case of the sandwich ELISA, you can screen the antigen, and it has no disadvantage, but it has the advantages: highly sensitive, flexibility and sensitivity, and suitable for the complex samples. Antigens are not required for the purification. While in case of the competitive ELISA, you can screen both the antigen or antibody, and it has the advantages that the high specificity, high sensitivity, and suitable for the complex samples. Antigens are not required for the specific that the purification. So this is the sketch of the prototype ELISA. The first one is coating, then incubation, then washing, then blocking, and then again incubation, then washing, then you add the conjugate, then washing, then incubation, and then you add the serum. And after the addition of serum, that you should incubate it, and then washing, then substrate, you need to add it, and then incubation, then stop solution, and you should take the OD value. This is the pictorial view, and this you can observe that this is the antigen, then antibodies which have the enzyme linked antibodies, and it show the color. So next is the protocol for prototype ELISA. Similarly, I have already described in the previous slides that coating, and coating should be with the protein concentration 0.5 microgram per ml or 1 microgram per ml, and you add the 100 microliter per well. In case of the incubation, then you put the plate at 4 degrees centigrade overnight, and then washing, and its washing should be done at that with 0.01 molar PBS and 0.5% 2 and it should be done 5 times. Then blocking, and blocking should be done with 4% BSA, and then incubation, it will be at the 37 degrees centigrade. Then washing, and after the washing, then you add the serum, then incubation, then washing, and after that you add the conjugate, and with literature cited that the ratio, and then incubation, then washing, then substrate you need to add in case of the AP linked conjugate, and incubation, that incubation is for 15 minutes, and then you should add the stop solution in case of the AP linked conjugate, that the 3 molar NOH, and then you should take the OD value. This is the ELISA. It actually, this plate show the positive or negative sera. This is the ELISA reader, and this is the curve that show the OD value. Or advantages of the ELISA: it has the less costly and safest and easily visualization of the result with high level of accuracy, and specific and high sensitivity assay that can detect the protein at picomolar or to the nanomolar ranges, easily automated for performance of the large number of tests, and require minimal reagents, qualitative detections or quantitative measurement of either antigen or antibody. Wells can be coated with antigen or antibodies, and can be done by personnel with minimal training. The application of ELISA: the analysis of the hormones, vitamins, metabolites, and diagnostic markers, and therapeutic the drug monitoring is also can be done, and diagnostic procedures for detection of infection. Then the limitation of ELISA. It is very expensive at the initial investment, and variable sensitivity and specificity of the variable test, and it shows the cross contamination. And protein calculations, in this protein calculation you can see that this is the calculated protein that 94.05, and you need to coat the ELISA plate with this concentration 0.5 microgram, while you need to total volume 10 ml. You put the formula and you need this concentration. Next, that preparation of 0.001 molar PBS, 0.5% 2 in 20, and PBS is you have already 0.01 molar, and you need to prepare this one. And total volume is 1000 ml, and you need to get the percentage of 0.05% in, and put this value in this formula, and you can get these values. So some basic concepts, the antibodies, also known as the immunoglobulin, and abbreviated as IG, and are gamma globulin protein that are found in the blood and are used by immune system to identify and neutralize the foreign objects, such as that the bacteria or viruses. And what is the antigen? It is actually a substance that when introduced into the body, and it initiates the production of an antibody. What is the immunoassay? A laboratory technique that makes use of the binding between an antigen and its homologous antibody in order to identify and quantify the specific antigen or antibody in a sample. What is the analyte? It is actually the sample that is being analyzed in the immunoassay, and the analyte is either it may be the antigen or antibody. Okay, thank you so much.